Gene Count SARS-CoV-2 Wastewater Kit. Concentration and extraction. Create wash buffer 1 by adding 60 milliliters of isopropanol directly to wash solution 1 concentrate bottle. Mix by swirling for 10 seconds or until any precipitates dissolve. Create wash buffer 2 by adding 160 milliliters of ethanol directly to wash solution 2 concentrate bottle, then mix by swirling for 10 seconds or until any precipitates dissolve. Rehydrate the vial of lysis supplement 1A with 6.6 milliliters of nuclease-free water. Mix intermittently for 1 minute by swirling. Do not invert the bottle. Label a 15 milliliter tube with identifying information for each sample. Add 6 milliliters of lysis buffer concentrate and 250 microliters of rehydrated lysis supplement 1A to each 15 milliliter tube. Then add 1 milliliter of wastewater sample to the labeled tube using a wide mouth 1 milliliter pipette tip. Invert 5 times to mix the sample solution, then incubate at room temperature for 10 minutes. After incubation, add 3.5 milliliters of ethanol to each tube, then mix thoroughly by gently inverting the tube 5 times. Ensure that the magnetic beads are fully resuspended by inverting 5 times before adding to the sample mixture. Then add 40 microliters of the magnetic beads to each sample tube. Invert the sample mixture 5 times to mix, and then incubate at room temperature for 10 minutes. Next, place in the magnetic rack to precipitate the magnetic beads on the side of the tube. Then carefully discard the supernatant. Add 1 milliliter of wash buffer 1, then cap and swirl the tube 10 times to mix. Place the tube in the magnetic rack to precipitate the magnetic beads, then carefully discard the supernatant. Repeat the previous 2 steps with wash buffer 1 twice more. Next, add 1 milliliter of wash buffer 2 to the magnetic beads. Cap and swirl the tube 10 times to mix. Place the tube in the magnetic rack to precipitate the beads, then carefully discard the supernatant. Repeat the previous 2 steps with wash buffer 2. Add 1 milliliter of ethanol to the magnetic beads. Cap and swirl the tube 10 times to mix, then transfer the mixture to a new 2 milliliter tube using a new disposable transfer pipette. Place the tube in the magnetic rack to precipitate the beads, then discard the supernatant. Remove the tube from the magnetic rack and allow the pellets to pool at the bottom of the tube. Next, place the tube in the magnetic rack and allow the magnetic beads to accumulate to the side of the tube for 2 minutes. Remove the remaining pooled liquid at the bottom of the tube with a new pipette without agitating the magnetic beads. Next, add 50 microliters of elution buffer NA to the 2 milliliter tube. Cap and gently swirl the tube 10 times to mix. Incubate the magnetic beads at 60 degrees Celsius for 5 minutes. Then place in the magnetic rack to separate the magnetic beads and use a pipette to transfer the supernatant containing the eluted RNA to a new sample tube for analysis. Open the RT-qPCR master mix bottle. Gently tap the bottle on a hard surface to collect the contents to the bottom of the bottle. Transfer 825 microliters of nuclease-free water into the RT-qPCR master mix bottle and let the RT-qPCR master mix rehydrate for 3 minutes. Mix occasionally by swirling, but do not invert the bottle. The rehydrated RT-qPCR master mix should be used immediately or frozen at -20 degrees for later use. Dispense 15 microliters of the master mix into each PCR strip tube. Then transfer 5 microliters of nuclease-free water into the first PCR tube. This is the negative control. Close the tube. Please note that only 1 set of controls is needed per instrument run, not per strip tube. Transfer 5 microliters of each sample RNA into the individual PCR tubes. Close each tube as the sample is added. Leave the last PCR tube with no RNA if adding an optional positive control. If using the optional positive control, you can now transfer 5 microliters of the positive control DNA into the last PCR tube. Close the tube. Please note that the positive control DNA is highly concentrated, so care should be taken not to contaminate other samples or your work area. Gently mix each qPCR reagent tube. Then, using a robust downward motion, shake the contents of the qPCR tubes to the bottom of the tubes. Be careful to note the correct orientation of the tubes to prevent accidentally reversing the tubes when inserting into the qPCR device. A small mark with a permanent marker on the side of the first tube can help prevent misorientation. Samples are now ready for analysis in the GeneCount Q8, Q16, or Q96 device.